Welcome to the Collaborative Research Center Origin and Function of Meta-Organisms, Lucía. It's a great pleasure. You are not a real outside speaker and guest. You're an alumni of the CRC. You were here for many years, and we are looking back with a very strong feeling of your important contributions here to the center. It's great that you are spending some time with us and thinking about more personal on your career, on your science, and maybe also on the future. And just before we start, I think you got your PhD in Barcelona. Then you went as a postdoc to Germany, to University of Würzburg, and you met there, I guess, a very important advisor, tutor, and peer, Ute Henschel. And then you moved with Ute from Würzburg to Kiel and to the Georg Helmholtz Institute, where you then continued as a postdoc researcher, and at this time you were affiliated with the Collaborative Research Center. And now you are a junior researcher and a group leader in Barcelona in the Institute of, I call it Ocean Science or Science Ciencia del Mar. Currently, you're actually in the US in a lab of a good colleague of mine, Virginia Weiss, and so we do the interview online from the US to Germany. Thanks for spending some time with us. And first question, you are a dedicated and enthusiastic symbiosis researcher. And how did you find this field, this question? Was there anybody in your life who pointed the finger out to sponges or corals or to symbiosis? Or how did you get to that?
I wanted to study the question that combine ecology but also molecular tools. So looking at the small scale but also the big scale of things. And I was attracted by the ecological concept because ecology acknowledges that the animals, they don't occur in isolation, but they interact with each other and with the environment. And I moved to the University of Barcelona. I did my master's studies there in the group of Xavier Turon, and they were starting a project in sponge symbiosis. So the topic fit a bit like perfect to me, because it was looking at the small things. I was looking into the microbial diversity in marine sponges, and at the same time, I had like this connection of an animal that plays a key role in nutrient cycling in the system, is very abundant, is important in terms of the biodiversity of the marine ecosystem. And this marine component was also important for me. I grew up on the coast. And at the beginning, when I studied my biology studies, I thought I would be going more in the direction of cell biology or microbiology, like finding out new vaccines, curing cancer, this kind of thing. But I did an Erasmus in France at a marine institution, and I love it there. And it connected very well to even to myself, to my roots. So I wanted to add this marine component into my career.
Thank you so much. This is just terrific, and the career went on very, very well. And if I can ask you just for our listeners here, summarize in a few sentences what, in your point of view, were the most important contributions and observations, the most exciting idea which came up so far from your research?
Well, one of the first points I found out during my PhD was that the diversity of the microbial communities within sponges is highly dependent on the species we are looking at. So even different sponge species in the same location, they will have a specific microbiome, and an individual of the same species, even if they are hundreds of kilometers apart, they share more common microbiome than with other species nearby. So this realization means that the host must be playing a role in controlling which microbes are inside. And that's how I decided to move into sponge immunology and looking at the immune system of sponges. And I was surprised how little we know. So recently, I've been looking at the sponge immunity from different perspectives in interactions with microbes, but also with the environment. And for example, in the context of wound healing, I was surprised to find that the response of a sponge to wound healing in terms of which genes are activated is highly conserved to what we found in other animal groups and even to certain processes in humans. And we are talking about animal groups that have been evolving separately for 600 million years, right? So this must be very important signaling pathways. And I think that was a recent new development in my research.
Thank you so much. Talking about sponge immunity, some people will ask themselves, immunity in sponges, how can you study that? And so an important question these days is, of course, not so much, do sponges have an immune system? But how can I address that question? The methods, the technologies, the instrumentation, the equipment. Is that something which you think is a major factor when thinking about which type of questions to address?
Yeah, for sure, this is a major challenge. It's not the same studying immunity on a system that is already established or in a model organism in an emerging model like sponges. But actually, we can say that the sponge immunology started with the work of Elias Mechnikov, the Nobel laureate in the end of the 19th century, beginning of the 20th century. So he was just using like a glass tube and inserting into the sponges, and he observed how sponge cells were aggregating around this foreign material. So it seems something very simple and truly an observation under the microscope, but he used this and the observation in other animal groups to describe cellular immunity. So I think we have like strong foundations and golden foundations, but then the field didn't develop so much. They have also, sponges have been used to investigate self of non-self recognition, because we can disaggregate a sponge and it can reaggregate again in a new individual. So I think now we have new techniques, and this topic has not been revisited. And we can use single cell genomics or single cell sorting under the flow cytometry to separate different sponge cells. We have, of course, molecular tools. And I'm also interested in establishing new microscopy techniques to investigate these phenomena, these cellular responses connected to the molecular responses of the sponge. So it requires certain development, but I think there are some simple experiments we can try to develop with new techniques and get new direction in findings.
When going through the list of the papers you have published so far, it's truly impressive, but also they have a real scope, and of course some of them with number of co-authors coming from very different disciplines. How do you cope with that? I mean, is that something which you think is the future, that you cooperate and you collaborate with people outside your own discipline, multidisciplinarity or interdisciplinarity? Is that getting important for your own research?
Yes, this is key for me. We can, if we work alone, we can only do a very limited amount of things. We need people from other disciplines to get not only new insights, but also new techniques and methods in our question. And in that sense, the CRC was a perfect platform, and it really changed my way into thinking into marine sponge symbiosis, because I had the opportunity to collaborate with people who work on model organisms, mathematicians. It was just, first, is exciting. I find it very exciting to share your research with others that are not necessarily from your field. And for example, now I'm in a lab, I'm visiting the lab of Virginia Weiss, who works on symbiosis, but on a different organism, on sea anemones. And it's also going a bit outside my own field, just to learn how these questions are addressed in a different organism. And it's really exciting. And then I come with new ideas to sponges as well. So it's very interesting.
I fully agree, and I think I can also understand you. But it's also very challenging, because I mean, you have to understand what the mathematicians are talking to you, and you have to somehow get the language. Is that requires some communication skills, I guess, and also the willingness to listen to the other languages, isn't it?
Yes, that's true. I think we need first like a common arena, like a platform or a conference where we can really encourage those encounters, because otherwise it would be maybe very difficult someone working at the faculty of biology to go to the faculty of mathematics. So certain platforms will already attract people that have an interest on the topic or a curiosity on what you are doing, and also having seminars at institution level will foster these interactions between departments. But then it comes to the point of how I can communicate that this is exciting, no? Why sponges, why symbiosis? I think we are lucky now in terms of symbiosis, because the word microbiome is in everyone's minds. It's in the news, so people are getting more familiar with it. And I think we just need to, if we are enthusiastic, we can also, people will feel that, and then we say, okay, we should try to work together. And in my case, it was never a problem. I could see that how your background will, then you come with different questions, and you cannot assume certain knowledge. I have sometimes a more interesting question coming from people very outside my field, because I take things for granted, and the people in the community, we take it for granted, and then you get this question that sounds very basic but makes you think a lot. So that's great. That's only encouraging more research and more interactions. So I think curiosity must be always a key component of any researcher. So in that sense, it should be easier for us to talk to other people outside our field, driven by this curiosity.
Interesting that you put curiosity and communication and listening to other people in the same sentence and in the same category. But we are here now already in the middle of your personal attitude and to science, and that brings me then, of course, to the question: since you are a role model, you got prizes here from the CRC, you got awards from other institutions, you are a role model, Lucía, and we are proud of you. And young people looking up to you, and they ask probably themselves, could I make it? What does it need? What does it take to be a successful scientist? And what would be your answer? You gave already some hints in your previous comments now, but what would be your answer to the graduate student? She's right now at the moment on a turning point and thinks, should I continue? And oh wow, I have talked to Lucía Pita, and what would you tell her?
So first, you need enthusiasm. You need to know, to have a passion from what you are doing. If you have lost that passion and you cannot find it again, maybe you should think about doing something else for a start. No? But then I think it takes, of course, to know yourself. What are your priorities? What you want to do, and also to know a bit the system. For example, if you love teaching, maybe you should not go to an institution where it's only research and there is not so much contact with the students. So I would consider all like multi-dimensions of a person. It's not only the topic, it's not only learning a new technique, it's not only where you live. So it's all together. And for example, for my first postdoc, I wanted to learn new techniques. I thought it was a good moment to get exposed to some different tools. I also thought about the lab I was joining. I must confess, it was also a bit of a gut feeling. Like I have this, like something pulling me towards, I want to go there. And then I was so convinced that you will find a way. If you are convinced this is what I want, there are different opportunities. It may take longer. It may be more difficult. Maybe you go around and you spend some time somewhere else. But if you get there, if you are convinced, sometimes what I see from the young researchers that it's difficult to choose what do I want. And for that, I would say talking to different people, also people in the group you think you would like to join, that always helps. And yeah, I also had great mentors and great advisors, and that definitely was a very important aspect in my career. So I could always trust that I could talk to them, and they knew me as well, again, as a scientist and as a person, so they could help me in my decisions.
Looking from here now back, everything is just terrific, admirable, impressive, what you achieved so far. May I ask you, I'm sure there were occasionally obstacles, challenges on your way. Was there maybe even a day or a night where you thought, that's now, I'm not sure if I should continue, or were there never any doubts? And obviously you overcame that all and you made it and you went on. So what about this more difficult times? I'm sure they are there in every career, and no career is straightforward. And at the end, all of that looks shiny. Okay, but there were...
Exactly.
Yeah. So...
yeah, Thomas, that's a great point, actually, because when we talk about career, as you say, everything seems bright and smooth, but there are also challenges around the way. And I can think of like 2 points where I was struggling. The first one, when I started the postdoc, I realized it was a big jump from the PhD to become a postdoc. It was also different dynamic. So it took me a bit of believing in myself to keep going. But I had great support, so it was not really a big issue. And then later, when I was deciding if I should continue into a junior group leader position at KOMAR or do something different, I also have many doubts. And again, it's because it's not all about science, and sometimes we are struggling for different reasons, and that may affect what we want to do or where do we want to be. And in that case, it was, again, a lot of thinking about a bit of personal work, like what do I want to do, what is important to me, talking to my colleagues, and and at the end I stayed, and I do not regret it, but it took some time and some energy and some of this more introspective work, like what do I want to do as with my career, and where do I want to go.
This sounds now as the real pathway, and that's very good. I mean, that's it's also good to tell about these more difficult times. I think everybody, also if I think back, there were crucial turning points where if I would not have got the right support or the right signal, then probably I would have done something different. And that's important that we realize that, and that we also then make the right decision after in these days. Yeah, that's excellent. We are living in very strange times. You were finishing your time here at Kiel University and at the GeoMar in the pandemic, and we still are not through yet. We are communicating at the moment here online. Otherwise, it wouldn't be possible at all. How did you cope with the pandemic? I mean, this is a sensitive time in your career, and then comes COVID, and you have to change drastically your normal academic and scientific routine. How did you cope?
So during the pandemic, I stayed mainly working from home. I had a lot of data and things to write, so it's not that the work stopped completely. And I think actually some of the effects in terms of maybe publication or so still come later. So the effects of the pandemic will be lasting longer than the pandemic itself. For me personally, I was doing well because I was feeling privileged. I could still work. I think the priorities changed a bit when you could see the situation, particularly in Spain. You know, the first wave hit very bad. So that gave a different perspective. But over time, I realized that my energy was completely consumed. It's amazing. I was working from home, just actually my living room, where I have a very nice desk, but it's still my living room. So, and I was not realizing, I was just trying to keep going. I applied for fellowships. We published papers. We had the students in the lab, so things were moving as usual, but it was just taking the triple of energy than normal. So at some point I have to to feel like I need to reset a bit and take care of myself so I can gather all the energy back and move forward. And something very funny that happened when I moved to Barcelona, that I have a bit of time to rest as well. Suddenly I was exploding with new ideas, and these are ideas I could develop in Germany as well, or 2 years ago maybe, but I didn't have the time to sit and relax, and my mind was busy with maybe noise or maybe worries. But yeah, so these are the silent effects of the pandemic, I would say. It's not evident because I was still working and things were happening, but there were also good opportunities missing also for conferences that are so important to establish new collaboration, get new ideas. Also for me as a young researcher, so people can also know me. Yeah, I think this kind of effect we will see maybe still in 2 years or so.
Yeah, for sure, I agree. But just listening to you, I realize you had actually an interesting and clever way of escaping some of the pandemic consequences, and that is you changed location, you changed physically, geographical locations during the pandemic. And when you tell us that actually caused a burst of inspiration and ideas and creativity, probably also due to that you had the gut to change locations, and that is then always changing locations and traveling and being in a new place is always very inspiring. And seeing that you are now in the US for a few months in a very nice lab probably also will be a lot of inspirational sources here. Yeah, good for you. We are coming to an end of this short interview, Lucía, but I would really like to ask you to, you're now starting your own group. You're a passionate symbiosis researcher. You are very active in communication and in social media, and you have very, very nice papers already. Where do you see your research in a 5-year or a 10-year perspective?
So I would like to continue working on a sponge immunity from a broad perspective. So how immunity mediates the interaction between sponges and the microbes, and also the role of the environment. And in the future, I would like to build a bridge to biomedicine and really try to find applied situations of what we learn on sponges. I think they are fascinating organisms. They have been very successful, and I want to learn how they happen, how they made it so successfully with the help of their microbes. And I think that we have a lot to learn. Maybe we can find new solutions to similar problems, and then try to find an application in the biomedical field or biotechnological field. So that's something that I really would like to do in the next maybe 10 years, and to understand better this system, not only for the conservation of these organisms in an ocean that is really facing climate change, but also to find possible solutions for that affect human health.
Excellent.
That's the dream.
That's the dream. It's very nice to have, it's good to have dreams, and it brings you back, I guess, what we studied this short interview is when you mentioned Metchnikoff, and I guess the old colleagues and naturalists mostly were medical doctors, and they studied phenomena always with in the mind what can we learn about regeneration and wound healing and well-being. Metchnikoff did a lot of things also with the microbes, as we all know, but always having the human and the patient in mind, and health and disease in mind. And so yes, I can fully understand that this is something in your perspective and in your mind where you want to include then or conclude your research. Thank you so much for sharing very personal views with us. We all appreciate that. And thank you for spending some time with us here in the Collaborative Research Center. Thank you so much, Lucía.
My pleasure. Thank you, Thomas.